All right, ready to take it away? Thanks. So I'm excited to be here. Appreciate the opportunity to talk to you guys. It's great we have such varied backgrounds here. So especially some marine biology. I'd like to hear your thoughts on my ideas as I present them to you. I do want to give just a little bit of background about myself before I get started. As an undergraduate student, when I was in their position, I worked on evolutionary questions concerning great white sharks and the megatooth sharks, so megalodon. For my master's, I went and worked with trilobites, and for my PhD, and now I work with clams, which doesn't sound as exciting about the other two. And honestly, starting out, I was kind of like, oh, okay, we'll see where this goes. But I think it's the questions, and hopefully I'll convince you of this by the time we're done tonight. But it's the questions that we can try and answer with clams, and particularly, the stories that are recorded in their shells. That's what's the most interesting part about this. And to me, that's what science is all about: asking and trying to answer interesting questions and seeing where they lead us. So before we really get started here, though, I want to talk to you about a field of research that relates to mine, and that is called sclerochronology. Right? So big word here, but relatively easy to understand field. It's the study of periodic features and the skeletal portions of marine organisms. Okay, so this is very similar to the field of dendrochronology, right? The study of tree rings, and that came first, and then the clam people and other people kind of borrowed lots of techniques and tricks from the tree people, right? Because you can look at ringed widths in trees, and they can tell you something about climate and a whole host of other things, right? Well, the sclerochronologists, they do this too, but they work with corals, they work with fish, and they work with, of course, bivalves. Now, all three of these are really good for sclerochronology because they have what we call indeterminate growth, right? They don't stop growing over the lifespans, and they grow by accretion. So they're slowly adding shell material, or in this case, this is an otolith, this is an ear stone. They're slowly adding that material over their life, and then we can go back and play around with some interesting scientific questions. A lot of sclerochronologists try to answer questions about climate, right? Because these organisms, as I'll show you, they are recording climate in these structures, and we can get at that using some isotopes, which is coming up here in just a little bit. But how have paleontologists used this, right? I am a paleontologist, and highlight as a biologist every now and then when I have to wear that hat, right? But I'm a paleontologist and a geologist. So why is a paleontologist interested in this field, and what have we done with it? So one of the earliest applications of sclerochronology to the fossil record were these studies by Wells back in the 1960s, right? And Wells looked at this thing over here, which is called a rugose coral. It's an extinct type of coral. It's no longer around. This one's from the Devonian of western New York, so about 320, 330 million years ago. And Wells noticed that if you look at these things, if you cut it in half, you can see these growth increments that look very, very similar to growth increments that corals have, right? And Wells argued that these increments were daily increments. And from that, he said, it looks like there were 400 days in the Devonian year. So the year used to be longer, right? And this is relating back to geophysical things and astronomical properties here, and how the Earth's rotation has changed over time. And the early days of sclerochronology and paleontology kind of, mixing fields here, this is kind of what they studied. So one of the studies that's relevant for my talk here today is this work by Barry and Barker in the 70s. And here they're actually looking at clams. These are clams that are cut in half, and they're looking at growth increments. And again, they kind of looked at the same stories here. They're looking at both modern and fossil individuals there. And they're arguing that the number of days in the year has decreased over geologic time, right? The year is essentially getting shorter, right? Now, these early researchers, when they're looking at these growth increments, they're inferring that these are formed tidally, right? So the tides change, and that stresses out the individuals, and they essentially kind of stop growing, right? And they record this little growth increment. And the early work really focused on tidal increments. And part of that is because it was very hard to determine which increments formed annually. That is until the work of Jones and others in the 1980s. Now, before this work, lots of people interested in lifespans of bivalves tended to argue that these lines that you see expressed here on the surface of the shell, that you could sort of count those to figure out how long that individual might have lived. But what Jones's work and lots of other people have showed us is that that's usually not true. Now, there are always exceptions to this rule, but most of the time, these lines that you see here on the surface, they're forming in response to disturbance events like storms, right? There's a really, really bad day, and the clam kind of stops its growth, it changes its growth pattern or something like that. So, in this study, to identify annual growth increments, Jones and others, they took these clams and they cut them in half and they sampled for oxygen isotopes. So, a quick aside about oxygen isotopes. There's three main varieties of oxygen on the planet today. Right? Oxygen 16 is your standard oxygen, and it's something like 99.98 something, I can't remember, percent. Right? That's most oxygen. There's also oxygen 17 and oxygen 18. And it turns out that the ratio of oxygen 18 to 16 in things like clams and potentially limestones, things made of calcite or aragonite here, it can actually tell us about temperature. Right? It's dependent on the temperature of the environment in which these things live. And so what they were able to do is they were able to take these individuals, cut them in half, and you can see those increments, and they're really pretty obvious in Spicula. And what they saw is a really, use a pointer here, a really nice sinusoidal signature which matched the temperature signature where these things were living. And it turns out that these little peaks here, they tend to match up with those dark growth increments. And then there, you know, these are annual, right? So now once you know that these things are annual markers, well, you can count them up and determine how long these individuals live. It turns out most Spicula from New Jersey live for about 20 to 30 years, which is pretty significant for a clam. But I'll kind of tell you a little bit more about that in a second. This is where we're going. Now, this is important because not only can we determine lifespan, but now we can start to think about things like growth rate. You know how long it lived, you know how big it got. We can think about growth rate, how fast it grow. So here's an example of that. This is a fossil clam. This is called Lahilia larsoni. This is really cool. It's about the size of a softball. It's a really neat clam. But if you cut this thing in half like I did here, you'll see really nice growth increments that are annual. You can count them up. This individual here lived for about 60 years. Now, to get a growth rate, what I'm going to use in my talk is this thing called the von Bertalanffy growth equation. And it looks scary, it's math, but really there are two parameters that you need to worry about, okay? The first one is this one that we call L infinity. If you notice this curve, and this is pretty standard for clams, this is how they grow. They go really fast in the beginning, and then for a long time they grow really, really slowly, right? They're adding shell material, but not much throughout most of their life. So L infinity is essentially an asymptote, right? It's a size that thing is approaching, but of course it never reaches. So it's kind of a proxy for maximum size. And K describes how fast the individual approaches L infinity, right? So if you have a high K, we can think of that as being faster growth. And if you have a low K, we can think of that as being slower growth. Now, it's not an absolute growth rate, but in a general sense, it tends to work. So after we've looked at the increments in this individual here, we've not only learned that this was very, very long-lived, but now we've learned that it's also very slow growing. Okay? So that's really interesting, but why does this matter, right? Why do I care about this? Why would paleontologists be interested in this. There's a lot of questions that I'm interested in and a lot of stuff that's driving my work. But one of the most important ones is this idea of how animals have changed their body sizes through time. So this plot over here on the right, this is from a paper that was published a few years ago. Got a lot of attention. It's a pretty big idea here. It's really messy. There's a lot of colors there. So the takeaway is this. It's showing you that there's an increase in the mean body size of marine animals over the past 540 million years, right? So things in the oceans really start to take off from the Cambrian explosion, and we go from there. And all of these groups, if you pull out individual groups here, you can see the similar trend, right? Things today are bigger than their ancient counterparts. And this works for bivalves too. Modern bivalves, on average, are bigger than their ancient counterparts, right? So from my perspective then, the question is, and I think this is a question that no one's really asking, or maybe not enough people are asking, is, well, how are they doing that, right? How are bivalves getting bigger? You can think of two ways to do it. You can either grow faster, or you could potentially live longer, right? So we don't know the answer to this question, in part because we simply haven't looked. We haven't done this, right? Now, this is a pretty big question. It's going to take a lot of time to answer. Right? We're going to look at 500 million years of bivalve life histories. It's going to take some time to get to. But before we start that road, start down that road, what we might want to know is, what do lifespans and growth rates of bivalves look like on the planet today? Like, what are we dealing with today, so that when we go to the past, we can kind of understand that better, right? Because we can go out and measure water temperature off, Hawaii, right? We can go to the Arctic and measure food availability and all these sorts of parameters. When we get to the past, we can do it. A lot of nuances there. It's a little bit trickier. So it gives us a starting point to work with. So this is the first real story here that I'm going to tell you. There are two stories that I'm going to talk to you about today. Now, as a PhD student, I was presented with this question. My advisor kept kind of pushing me in this direction, took me a while to figure out if that's where I needed to go, sort of thing. Sometimes that's how it works. But, how do lifespans vary on the planet for bivalves? And at first I thought, well, someone's got to know the answer to that. That's got to be out there, right? We know a lot about bivalves. People like to eat them. We might want to know how long they live, sort of thing. But once I started digging, it turned out, well, we really don't have, or we didn't have, a good answer to that question. So what I did to answer it is I got a bunch of colleagues together over here. There's me, just to not have a beard. My advisor and some both undergraduates were involved in this, and graduate researchers at Syracuse University, where I was a PhD student. And we set out to do systematic searches, right? So we started with a list of 30 journals that we thought might be appropriate, might have lifespan or growth rate data in them. And we went through every volume and every issue and looked at every single article. This is why I got undergrads involved. Every single article in it. And every time we found something that talked about lifespans of bivalves, we reported that information. And then we tracked down every source that was cited in there that might have information in it as well. So you can start to spiral out here and get lots of data really, really quickly. So at the time, this thing is published as a citation on the next page, but we ended up with 670 articles with data from 216 journals. There are representatives from just over 1,100 populations around the planet. And we've got representatives from 45 families, 158 genera, and 298 species, which is a lot. But when you think about how many bivalve species there are, or there may be, 20 to 30,000, depending on who you ask, we're scratching the surface here, right? But we gotta start somewhere, right? This is what we could find. So we recorded information on maximum lifespan. What was the oldest individual from that population that was reported? Size using L infinity, and K, we use that for our growth rate, right? Then when it was available, we looked at depth and other things, but it was really kind of spotty. So these were the main things we were talking about, and that's what I'll focus on in the next couple of slides. So let's first start with the question, how long do bivalves live? Well, it turns out that most bivalves are actually relatively short-lived, as shown here in this histogram, right? The modal age in the database is 3 years. But there are several species that live for 50, 100, or perhaps even 500 years, right? So let's take a look at a few of those. Mercenaria mercenaria, which is really common along the Atlantic coast, has a maximum recorded lifespan of 109 years just off of Cape Cod. Right? Pinopia hinerosa, 168 years from British Columbia. Lysimachus glycymeris, 192 years from Scotland. And finally, Arctica islandica, 507 years from Iceland. And that is the record for the longest lived non-colonial animal on the planet. It's a clam, 507 years. And fun story, that one was actually alive when they collected it, so they killed the oldest living animal on the planet. But they didn't know that. They didn't know, you gotta think back to that growth curve. They didn't know it was gonna be that old. It was unfortunate, but that's what happened. So, as a paleontologist, one thing that pops into my head is, well, all of these, I've seen all of these in the fossil record, but no one's bothered to look at what their lifespans are. So there's, work to be done here. But there's another story here that's coming out, and maybe you've picked up on it, right? The shortest lived one that I've showed you comes from about 40 degrees latitude, and the longest lived one comes from 65 degrees latitude, right? So maybe there's a story there. Maybe there's a trend. Of course, I could have just cherry-picked the data, and we could stop right there and go home and not worry about it, but we actually have the data now. Look at this. We recorded everything by latitude. So let's take a look at the relationship of latitude versus lifespan. So sorry that popped up. I was supposed to wait on that. But over here on the left, we've got absolute latitude. So we're northern and southern hemisphere. We're combining all into one here. And we've got lifespan on the y. Right? Now, realize that lifespan, the y-axis, it's log scale, so you got to think a little bit differently about how that trend is going there. And when we take a look at that, what we see is that tropical bivalves, guys down here, well, they tend to be much shorter lived than mid and high latitude bivalves. So the pattern I was kind of describing before, it seems to hold when we look at bivalves as a whole on the planet today. Now, if we look at growth rate, and using von Berg K as our proxy, right? Remember that lower K values are slower growth and higher K values are faster growth. We see a similar, but it's an inverse pattern, right? Those tropical species that are shorter lived are also faster growing. And then as you go up to the longer lived, higher latitude species, they tend to be much slower growing, right? So this is interesting, but what's controlling this pattern, right? Well, if we think about the community that studies the aging process, and this is where I have to kind of put on a biologist hat, and hope I don't get yelled at too much here. But people who study aging, there's a lot of debate on what controls aging, and that's a whole different field here. But one of the most agreed upon factors is metabolic rates, right? And it's pretty well agreed upon that animals that have higher metabolic rates, well, they tend to live shorter than those with lower metabolic rates. So maybe that's playing a role here. Now, there are ways you can actually influence the metabolic rates of organisms, and we have done experiments to show this, and there are data to support this. But there are two big factors. One of them is temperature, right? So you can think generally of metabolic rates as like the summation of chemical reactions in the body. And most chemical reactions proceed faster at higher temperatures, and it seems to work the same way with metabolic rates. So maybe the faster growth rates of those lower latitude bivalves, maybe it's just driven by warm temperatures, right? Now the other way to play around with metabolic rate here is to restrict food availability. Right? People have actually done work on this where they've introduced what you call caloric restriction. Simply just don't feed things for a while. And it tends to slow down metabolism, and it actually promotes longer lifespans, and it has all these kind of aging benefits. Now, I'm not telling you to go out and starve yourselves. Don't take that away from this talk, right? But there is some research to show that maybe this Now, our question then is, are those slow growth rates of polar bivalves, are they driven by seasonal availability of food? Now, what I'll tell you later on in this talk is that food for a bivalve is in part dependent on sunlight. Think about the sunlight regimes at polar latitudes, that influences food availability. So maybe there's something going on there. Now, the issue is, if we try to look at these two together on the planet today, it's kind of difficult to separate the influences of these, and that's because these co-vary with latitude. If you move up in the latitude today, it gets colder, and there's less food for certain parts of the year. There's seasonal availability of food, I should say that. Now, thankfully, this is where I become a geologist, right, and say, well, Earth's history has changed a lot, right? The planet always hasn't looked like this today, so we have all these wonderful settings where maybe this isn't, in fact, the case. Maybe the poles aren't always cold throughout geologic time. And that takes us to the second story that I want to tell you today. And that is going to take us down here to Seymour Island, Antarctica. So you're all probably aware of what Antarctica looks like. It's pretty cold there. There's not a lot that grows there. It's kind of barren. And it's not somewhere you want to live for a long time unless you're a penguin, you're pretty well adapted, or some crazy scientist who's got some money to go down there and poke around for a little, right? But of course, it hasn't always looked like this, right? About 40 million years ago, that's what Antarctica looked like. So notice, I feel like every time I look at this, something else pops out at me, but I'll help you out here. Okay, that's not an Antarctica today. It's kind of mammal, maybe it looks like a possum, really, is probably what it is. Elephant-like creature, I'll say ostrich or emu, a large bird. That's an angiosperm, a flowering plant, and those are coniferous trees. You will see none of these if you go to Antarctica today, right? Very, very different 40 million years ago. And in fact, temperatures in Antarctica at this time, we think were similar to modern-day North Carolina, right? So this is somewhere you might want to go visit, back in what we call the Eocene. Now, we have to also, I should mention here that Seymour Island today, there's a tip of South America there to orient yourself, right? We're looking up at the globe here. It's at 64 degrees south latitude, and we think it hasn't really moved much since this time. It's pretty much in the same spot, maybe a degree or two, depending on who you ask, but it's not like this was at the equator and then it's just moved down that fast. No, it was here, right? So it's down south, it's really warm, it's very, very unique. Now, relevant for my talk, of course, is what's not shown here off the coast, where all the clams were living, right? Which are now recorded in the rocks in Antarctica. So, what we knew about before I started this work was that there was one individual here, this thing called Cuculea rea, and that's actually it right there. There's a whole bed of Cuculea, which is probably another interesting story that we don't have time to tell, but we knew this one, people had already cut it in half and look at these growth increments here. We knew it was living for a long time. Several individuals, probably over 100 years. Now, this is going to allow us to test hypotheses of, is it food availability or is it temperature at high latitudes that's promoting long lifespans and slower growth, right? Because this is a warm, high latitude setting, which doesn't exist on the planet today. So when we look at this, we kind of set up the hypothesis here is that, if the environment is controlling longevity in some way, then you should see more than one thing that's really long-lived, right? It could be that maybe it's just Cuculea. Cuculea is really weird, and it's figured out how to live for a long time. But if we see other long-lived individuals, then that's would lead us potentially to think maybe the environment's playing a role in extreme longevity. So to test that idea, how do time here? Okay, I went and visited a couple of museums. I wish I could tell you I got to go down there, but I didn't. We tried to get funding, but of course that's hard to get. I went to a couple of museums: the Florida Museum of Natural History, the Paleontological Research Institution, which is in Ithaca, New York, and we had some on hand at Syracuse from the My advisor had gone down there. And I picked 10 different species that I could look at. I'm just showing you a few of them here so you get a sense of what these things are. And I picked these 10 for a couple of reasons. One, they were the common components of the marine fauna. Right? I'm not picking anything weird. I'm just kind of picking the things that are abundant. And two, what I've hinted at but maybe not explicitly told you is to look at those growth increments, I've got to cut these things in half. Now you can imagine walking into a museum, especially from hard to get to places like Antarctica. Right? You need big enough plants to get down there. So these were kind of the ones that they would let me take. Right? After some begging and negotiating, this is what I could get. So I got 10 of them. And they cover, let's see, 3 different families and 5 different genera. So we got a lot that are what we might call as unrelated. Right? We're getting lots of different species here. Now, because there hasn't been a lot of work done on looking at internal growth increments, we played around with a couple of different methods of visualizing them. So let me show you those here. So in this first method, we just took the clams. This is Lohilia largina. You actually already saw this species earlier. And we cut them in half, and then we just polished them, right, with a lapidary wheel. It's got silicon carbide disc on them. It's like sanding. I essentially sanded a bunch of clams, right? And you can see these really nice annual increments there, right? So that one goes up to about 30 or 40. I think I did every other one there, right? So that's pretty nice. We also made what we call thin sections. Okay, so you can take these things and make a thick section, and then you cut it down to 30 microns, which is really, really thin, and it allows light to pass through it. So in this case, we're using a petrographic microscope, and we're looking at light that's been transmitted through the sample. And it gives us just some different details that we didn't have before. And we can see, again, we've got really nice annual increments. We've got some intra-annual stuff that some of you guys up here in the front might be able to pick out, which is kind of food for thought later on. There might be something going on there, some more stories that we can tell. But the takeaway, nice annual growth increments, right? These are really easy to count. It's really nice. The final method is we use this stuff called Mootvi solution, which turned the thing blue. It's a dye, and it works with some of potentially the organic material that's still left in these things. There's not much there, but it binds preferentially to that, and so it creates differences in staining. It just increases some contrast here. We didn't use a whole lot of this in because, honestly, I was a little worried about working with it. It's got some carcinogens in it. We need to make sure we know what we're doing. I use a lot now. I train students to do it. It's safe. Figured it out. We know how to do it now. And it works pretty well with fossil specimens here. So again, you can see really nice annual growth rates. Okay. Now the takeaway from all of this, right, is this is just me convincing you that I can do this. I can count annual growth increments in things that are 40 to 60 million years old. They're there. They're visible. And I'm not going to show you the other species we looked at, but hopefully you'll trust me and say, yeah, they probably all look like that, and we're good to go. Okay? So I told you I was limited in the number of samples I was able to get. I was able to cut 34 individuals from this farm. So let's look at the results. All right. Of the specimens I examined, I want you to look at right here, maximum lifespan, MLSP. 131 years was the longest lived individual, and 22 years was the shortest lived individual. Now think back to that histogram of lifespans that I showed you for modern bivalves. The modal age on the planet is 3 years today, right, from our database. So 22 years is actually pretty significantly long-lived for a clam. So we would argue that all of these taxa are very long-lived. Now, if we look over here at the K values, what we also see is that they range from 0.5 to 0.29, which is actually relatively low. So not only are these Seymour Island taxa long-lived, but they're also very slow growing. So let's revisit our first question here at the beginning of this story. What factors influence lifespan and growth rate that are essentially related to metabolism. Remember that Seymour Island at this time, it's a high latitude setting, but it's very, very warm. It's like modern day North Carolina. So it can't be cold temperatures. We can rule that out, right? That can't be the main factor that's promoting long life. Food must be playing a role too. So some review for us here. I'm sure we all know, given our backgrounds in here, that sunlight varies a lot on the planet today. In part because of the tilt of the Earth and the fact that Earth is a sphere. It's not flat, it is a sphere, right? We have 6 months of day in the northern hemisphere, the pole, during summer. At the same time, you get 6 months of light. So you have, at the South Pole. So really extremely seasonal light regime here. Okay, what does this have to do with the bivalve? Well, let's look at this one more way here. Let's look at it graphically. You can actually download sunlight data from NOAA. You can get lots of data from NOAA. It's really cool. So I just pulled these, right? You got 10 degrees south here. That's the red line. It's relatively flat, which is you guys probably, expected there. And if we look at 64 degrees south latitude, well, it's extremely seasonal. From 20 hours of sunlight in what would be the southern hemisphere summer, right, to maybe 5 in what would be winter. So what this means for bivalves living in these conditions, right, if we think of food for bivalves, most of it is, let's say a large part of it is phytoplankton that they filter out from the water column. Right? Now, lots of bivalves are what we call filter feeders. Not all of them are, but the ones that I'm looking at here are filter feeders. So they're taking food particles that are floating around the water column. They inhale them through these incurrent siphons, and then they can exhale them here, right? This is a common muscle, right, to illustrate this idea. So what this means is that at low latitudes, food supply is going to be relatively constant. You just got about the same amount of food all year round. They're kind of happy. Now at high latitudes, food supply then is going to be extremely seasonal. Right? There might be several months of the year where there's very little food available to them. Right? But then a couple months, things will be great. There'll be lots of food. But for most of the year, there's not going to be a lot going on. So what I talked about at the beginning of this section is that caloric restriction has been shown to promote longer lifespans in some animals and promote longevity and decrease the effects of aging on lots of individuals. So maybe what we're looking at here is that slow growth, which would be affected by low and seasonal availability of food, that might be a variable to which long life could be selected for, right? Particularly at these high latitudes. So these findings shed some light on factors that influence lifespans in bivalves and potentially other organisms, but I think they have some implications, significant implications, for the history of life on our planet. So let me talk about these real quickly. Now the first, and this is a hypothesis. This is a testable hypothesis. It's going to take some time, but we could do this. High latitude settings should be characterized by extreme longevity throughout Earth's history, regardless of what the climate is doing. We should typically find long-lived things at the poles. So there's not a lot of work that's been done on lifespans of fossil bivalves, but some super anecdotal evidence here is that in the Eocene, we've got subtropical bivalves living for about 5 years. In the Jurassic, mid-latitudes, about 20 years. And in the Permian, I'm going back down in time here, high-latitude things living for over 30 years. So maybe there's some weight to this statement. Maybe it holds up. We'll see. Now, the second one, and this is something I really haven't introduced you to before, is an idea I'm thinking a lot about, and it's maybe that modern and ancient latitudinal diversity gradients. And let me just mention real briefly, if you look at the number of species in the tropics today, it's much higher than the number of species at the poles, right? Species diversity decreases as you move up in latitude, and that holds in most groups, right? And we think that might hold in the fossil record too. So maybe that pattern is driven by the pattern of decreasing growth rate with latitude. And that might be because taxa with shorter generation times, they actually can accumulate more variation at a given interval of time. They're actually think of it as like making more mistakes in a smaller interval of time. And also that slower growing bivalves, well, they tend to delay the onset of sexual maturity. Antarctica Atlantica that I showed you earlier in Iceland may not be sexually mature until it's 15 to 30 years old. Right? So that's a long time to wait to reproduce. So that might make polar species less likely to spin off new taxa. Right? There's maybe less chance for evolution to occur there, or selection to operate. Now, let me just summarize real quickly what I've told you, because I've talked way longer than I thought I would. It's okay. We know today that there's a latitudinal pattern in lifespans and growth rates of modern marine bivalves. And I think that maybe this trend in body size through time that I showed you, it might be driven by an increase in K through time. Maybe things are growing faster. Right? They're probably not living longer. I think they're probably growing faster. And I've also shown you that there are really impressive lifespans and slow growth of fossil bivalves from Seymour Island, which is this weird non-analog warm high latitude setting. And that suggests that maybe it's the seasonal availability of food at high latitudes, not cool temperatures, that is a driving factor in extreme longevity. So with that, I will stop talking, and I'd be happy to answer any questions that you might have. Thanks. Yeah.
The data you showed us looks like it comes from intertidal and shallow subtidal bivalves.
Yeah. So depth is, I would say, it's mostly subtidal. We do have things that go a little deeper. We're still on the shelf with most things. There's one weird, bizarre thing that's way down deep. But yes, you're right. It's mostly relatively shallow.
Well, the extension of that question is a chance to look at that or think about that and see what.
I haven't. Yeah, just because I can't get them. But if someone has them, I would love to look at them. Okay. You never know what someone has laying around in their garage. No, seriously, you'd be surprised. And there may be, there's this little hint, and we got, when we published this paper, we got this question, and it's buried in the supplemental data. There's a hint that there might be a trend with depth to where these deeper things are living longer, but the way people reported data in these studies is extremely frustrating. For example, there's one that I can remember goes from we sampled from 10 meters to 100 meters. Okay, there's a lot that can happen in that range. So from our data set, I can only say, I can speculate that there might be something there, but I don't know.
What would you expect? Would you expect the delta T of the environment to, if it's very small, would your growth rings be a lot less apparent? Would they be non-existent?
Well, I think that could be the case. It may be trickier when you go down with depth, because part of why these Seymour Island things, why those growth rings or increments are so apparent, is that extremely seasonal environment. Right? It's shutting off. Things aren't happening for a couple months out of the year. Now, is that going to be expressed in deep water? I don't know. I would think not. I got some oceanography, you have an oceanography background here. It's gonna, temperature's gonna stable off once you get below a couple hundred meters. It's all about the same. Yeah. So I don't know. I don't know the answer to that. And what the workaround might be is, well, maybe you can't look at increments, but if these things are still growing, you could potentially just sample the hell out of it for isotopes. If you got a lot of money, and maybe you can extract that record and kind of see if you can see something, but it's going to be tough.
Have you been able to correlate your isotope ratios with actual temperatures?
People have. Yeah, that modern study, it lines up really, really nicely with temperature, especially in shallow. Yeah.
Back at the beginning, you showed that big colorful plot from the other people about growth rates over the millennia.
Which back?
Back, yeah. Maybe that one.
Oh, that one. Yeah, it's body size.
That
Yeah,
body
yeah,
size, yeah.
It's noisy.
Very noisy. Did they separate it out? I mean,
They do, and
mean,
I actually
like it looks to me like arthropods actually get smaller over time, and
yeah,
and mollusks, it's not clear. They seem to
It's not
they seem to
clear.
diverge maybe.
yeah. If you look at the groups, and this is where I took out. I have way many more slides than I have time, but I pulled out the bivalve data from this, and it is increasing in bivalves. It's there. I think it's real, right? Now in arthropods, I think you're right. It looks to be decreasing, but you can also we can try to explain that, right, and think about what happens around here. People who took historical geology with me, evolution of insects, right? So you start to get all these really small things and farming plants, right? So that may be pulling that down a little bit. But I think in bivalves, I will say in bivalves, I think it is it's there. Just send me the plot.
Okay. Yeah.
And I'm saying,
Yeah.
this is obviously not my field. I probably eat my share of bivalves, but don't know much about it. But I'm sitting here thinking to myself, you had mentioned briefly that we eat them,
Yep.
right?
Yep.
And smelling the pizza, since you're saying that. Then I'm thinking, okay, I've never thought when I'm eating one of these, how is how people eating these things? Now you're looking at a long period of geologic time, so it probably just scratching the surface, but I'm thinking, how long have we been eating them as a species, and how the production of them, has that changed in any way? Could it change
Yes.
what you're seeing?
I think that's a really, really good point. Okay, so to try to answer parts of that question, we've been eating them for a very long time. There's evidence of, I'll just particularly speak to North America. In the archaeological record, you can see shell middens, right? And they can go back a couple thousand years, especially along there's some really good ones along the Atlantic coast, right? And there is a paper out there that tried to, so a shell midden is they go out and grab these things, in really shallow water, right? They eat them, and then you got to put it somewhere. Well, we just kind of make a pile, right? We're just piling these things up, and they've tried to look at how are growth rates and lifespans changing throughout that midden. It's tough because things get kind of mixed up. It's not a perfect layer cake kind of type thing. And there is some suggestion that maybe even Native American communities could have had an impact on how these things are growing. Now, to further illustrate this question, and it's really good. A lot of the data that we have here, we have because we eat a lot of these things, right? So we got to know how long they live and how fast they grow to manage a population, right? And in the U.S., the shell fisheries industry, early 1900s, they start picking up. And in the 1930s, 1940s, they really pick up again because we need a source of protein, partly for the war effort in the 40s, right? And then after that, it really picks up, right? So we've been going after these things for maybe 100 years with kind of modern technology sort of stuff. So I think we've probably had an impact on some of these guys. And I pull up this one to illustrate this because this is really important. Mercenaria. If now it may be different here, but if you're on the Atlantic coast or if you're maybe in Texas for our students over there, if you go and you get steamed clams, probably going to get Mercenaria, right? And they kind of farm a lot of these today. Aquaculture, very similar to what they're doing here with the abalone, right? You can pump food and stuff in there and make these things grow real fast, get them to market quickly, and they're viable. But with Mercenaria here, I think the data are all screwed up, and I think we don't really know how long they can live. The study that found the oldest lived one, they just went into a little bit deeper water, like 10 or 20 meters deeper water off Cape Cod, and they found a bunch that were approaching 100 years. Where close to the shore, where we've been harvesting them for much longer, 20, 30 years, right? So I think we have to be a little careful here because there are definitely some nuances in our data. And then to add on to that, if I showed you a map of where all these things are coming from, it's the Atlantic coast, it's Europe, We've got some from Japan. I mean, we have a decent global spread, but there are definitely hotspots. Right? And this is what you face in any kind of, when you're trying to do these global studies, it's where people have been studying them for the longest. That's where the most data are. Right?
Which in itself is an impact on the questions that you're asking.
Of course. But I still think, and in that paper we do a bunch of, send it to you if you want, John. We do a bunch of resampling to kind of prove that, this isn't random, right? We can, test what it looked like randomly in our pattern. We're pretty sure it's real. But it has definitely been impacted by humans. What I'd love to do, I'll stop after this because I'm giving you a long-winded answer here.
If I have a not question, you can talk to me more about it.
Yeah, what I'd love to do is go into the museums and get the samples from the 1800s, and we have them. The Smithsonian has them. I thought I was going to be able to get them, but that fell through, and I'll leave it at that. To see what populations looked like before we started harvesting them, because I think that's fascinating.
I enjoyed it.
Thanks. I'll try to make
Have
it
you
short.
or can you look at fossil species, and do you think that the fossilization process is going to change with the data you got? Is it going to change the isotope ratios, for instance?
No. Well, it can. Fossilization definitely can, and there's a bunch of ways you can test and see if those isotopes have been altered. Basically, if they've been diagenetically altered, and I don't do this, so I'm blanking out on this term, but there's some microscopy you can use to look at to see how the crystal structure might have changed, and if it's potentially changed, then you need to be leery of your isotopes.
Have you looked at fossils? You showed that bed in Antarctica, those
Yep.
clams, were they fossilized? Or?
Yes. Those are fossils. Those are 40 million years old, and those people have done in that study I showed you, the first one where I introduced that. They went through the steps to say, yeah, those isotopes, that's probably what it was. Right? And we're pretty confident in that. There's a lot of filters you can use, but it's tough sometimes. I've looked at fossils from the same locality, one from here and one from here, and I can see growth bands really good in this one, and in this one, not so good, right? So there's some more stories there, I think, to be told about how that process alters growth. Definitely.
Is there any way to do any non-destructive sampling of the players?
Yeah, you sound like a museum curator. Some people have asked me about CT scanning, and I just don't know enough about it, and I've never had access to a CT scanner. There's these micro CT scanners that you can do. It'd be fun to try. I don't know if it would work. It'd be great. I would guess, and I'm just guessing, it'd be really cost prohibitive, right? It would probably cost a lot, because to process these, I mean, it's pretty cheap.